it's my honor to introduce Alice. So Alice has started her journey back in France in University of Angers, where as a plant biologist, and then through her master's, where she moved from plant biology to more bioinformatics, and then for her PhD, she moved to Switzerland, where in the lab of Daniel Kroll in which is evolutionary genetics lab in University of Neuchâtel in Switzerland. She's studying the comparative 3D genome architecture in fungi. And she's right now wrapping up and finishing her PhD and searching for positions for after her PhD. But we are excited to hear what she has done for her PhD project. And yeah, Alice, the floor is yours. Welcome to Fragile Nucleosome.
Thank you so much. So first of all, you're going to see some tiny images like this. They are watercolors that I did during my project, and I think it's going to be great. So it's going to be my first author article, and I'm going to share it with you. It's such a pleasure. So my topic is about comparative 3D genome architecture in fungi. And why is it important to learn about it? It's because it's linked to development, but also like disease and cancer, but also like stress responses, whatever if they're biotic or abiotic, such as temperatures, but also microorganisms. And also it's linked to speciation and adaptation. So people have worked a lot this last decades on how chromosomes are interacting, and there is an amazing paper that tried to do some configuration, like to categorize the chromosome architectures. And so in animal and plants, you have, especially in animals, you have like the chromosomes that are going to much more interact with themselves than with the other chromosomes. And what is red here is what is interacting the most together. So this is called chromosome territories, and you will hear about it later. But also we have like the rubble configuration where you have the telomeres at one side and the centromeres at the opposite in the nucleus. And inside the rubble configuration, you also have subgroups, which is like telomeres and centromere clustering. So there you're going to see different maps that are going to interact much more at these very specific points. But I'm not going to go into the compartments and details of TADs and loops. It's not going to be my work with this paper. But so fungi are very understudied because we have, so what we found, like what has been showed is that in yeast and rust, for instance, you're going to have the rubble configuration. But what is also super cool is that in some fetal pathogens, you're going to have core and accessory chromosomes. And while the core chromosomes are interacting together, the accessory chromosomes are going to be apart and differently organized. But why is it important to work with fungi? It's because they represent so much species. So they have been estimated at 2 to 11 millions of species. And for now, only 150,000 taxa has been described. So I'm not even speaking about this 3D architecture, right? It's like about like 200 data sets, but we comprise this with a lot of yeast and a lot of strains, not necessarily the number of species studied. So comparative analysis of 3D genome architecture is really recent. It has been mostly done in animals. There are like actually very cool studies in butterflies and more incoming, but it's really like evolutionary restricted to animal and plants mostly. So my big question is, what is the diversity of 3D genome architecture and how did they evolve? Obviously, I'm not going to answer this question today, but we try to explore the 3D genome architectures in fungi and what we need still to understand and what has to be, what are the works that could follow up. So fungi are very good models in the way of they have very small genomes and they can be very well assembled, but also they have a very fast evolution in terms of genome expansion. So I try to find the best and the most robust data set, and that thanks to the Darwin Tree of Life, which is like a consortium in UK, they are trying to assemble all the genomes of eukaryotes, and they are doing an amazing work. Honestly, I'm so grateful for that. And so they had already quite some fungi in their data set. And so the one that had high C, I had 54 species, sorry. And I also added, so in three different taxa, which are like Mucoromycetes, Ascomycetes, and Basidiomycetes. And I also added one species, which is Rhizophagus irregularis, because I had only one species in Mucoromycetes, and I did not want it to have a blurry expectation about it, while if you have a second one, at least you can cross and see if it makes sense or not. And then we also added for the three-eyed group species, which are part of the Pagomycota group. But you're going to see these three quite often. But what is really nice is we have such a diversity among them. For instance, you have like some pathogens. Like here on top, you have one that is like a pathogen of plant, but you also have like a pathogen of fungi down with a Volvariella surrecta. So I think it's really nice. But you also have symbionts. We also have yeast, by the way, and marine ones, but we also have saprophytes. So we have different taxa, but we have also like a lot of trophic modes. And it's really cool to see if there are links or not. And if we can understand if the 3D genome architecture is linked to whatever, if it's phylogenetics, genetics, or like trophic modes. So what is the diversity of the 3D genome architecture? So first, I'm going to explain a bit this, because you're going to see it quite often in the next slides. So on the left, you have a 3D model where each bead represents 10 kilobase pairs. And the color matters in the way of more it's going to be red and more it's going to be the longest chromosome, and blue is going to be for the smallest. And on the right, you have exactly the same data, but visualized in a different way, which is the common high C map that we see with a high C basically. So I'm going to show you a bit of diversity of what we have in our data set. So what you can see here is like the shape is going to be different, but also the patterns that you can see in the high C map on the right. And I'm going to like discuss later how we try to categorize them and how we can find some links between phylogenetics or not, and what's the impact with the genetics as well. So first we found 10 rabble organizations in our data set, such as this one. Yes, it's working, it's nice. But this was more than expected, since almost only rabble organization was found in fungi for what has been previously published. But what was quite interesting is also that we have a chromosome territory species in the Ascomycota. So this model is a bit weird with the red shape, but it's just because the chromosome 2 is not as well assembled as the rest. That's why, but it's just matter of model. And then we have this kind of weird shape of bean, and it's really specialized and only found in Basidiomycota, but also especially in the Russulaceae family, which comprises Russula and Lactarius genera, and we had like 10 species out of them. But if we had 21 species that could fit in the three different main models, we had a lot of gradients. We have like intermediates everywhere. And we have actually one species that is between the chromosome territories and the bean shape, which was quite surprising. And it looks like this, just down of the triangle. But I would like to focus on this one between the rubble and the bean shape. You have some very compact bean, let's say, where actually you have you can see it by model and also in thanks to the heat maps, but you cannot see it in microscopy. So I think it's very important to cross the different analysis when we study 3D genome architectures, because if you have a round, you're not going to see how much like the chromosome are stretching. So I think we need to cross the data all together and find like different techniques to study the 3D genome architectures. So we tried to see how much like the 3D genome architectures were correlated with genome features or not. So on the left, you have like a schematic representation of the tree, and you have also, again, the models, how we define them, and you have after the genome size and colored by GC content. But what we found or did not found is there is no correlation between the 3D model and the genome size. And we tried a lot of different things with chromosome number, etc. But what we can discuss is about the T coverage, like mostly T content, sorry, and how much they are interacting. I mean, what is the impact, for instance, with genome size? And as expected, it's correlated with, so T content is correlated with genome size, but also it is not correlated to the 3D model. So I'm not going to speak about more about all the correlation and regressions that we worked on, but you will be able to see it in the paper. But mostly 3D models is not correlated with almost all the parameters that we looked at, except the tDNA elements. So the take-home message for me is that we found a new configuration, which is the bean shape. You could also like speak about the shell shape, but we went for the bean shape. And it's not only an artifact. I was very scared at first, but we found this paper about a Basidiomycota that has this very weird shape, so the bean one, and it's not only an artifact, it can be happening in real biology and not only simulation of heat maps, right? But also we found the chromosome territories in fungi, which is more than surprising since the recent paper of Owen Camp and colleagues showed that if you have a loss of some condense in two subunits, you're not supposed to have the chromosome territories, while and the fungi are not supposed to have it. So the question is, did this fungus got horizontal transfers, or is like another mechanism? We don't know yet, but I think it would be super interesting to dig into it and to see why at least one species has this configuration. And I'm pretty sure that more we are going to see. The thing is, I really believe that we need to sequence more and that we cannot see the chromosome configuration only in very stringent boxes, but much more as a gradient. And I think we have a lot to discover. And for the last minute, I'm gonna thank a lot of people. So basically, I'm really thankful for all the people I meet in Neuchâtel, but also like to this Wellcome Sanger Institute that did an amazing job at sequencing and assembling the genomes. But also I have, through my journey, met a lot of people, and these are names of the people I can think of all the people that supported me. You have the names of my mentors as well, but also my friends and family. And I would like to thank you as well. Da-da-da-da.
Thank you, Alice. That was a great talk and so on time. Um, so anybody, if you have questions, you can write it either in Q&A or raise your hand and I will unmute you and you can ask your questions. Let's see.
I cannot see the chat though.
It says in the it will be in the Q&A box if somebody has a question. In the meantime, I have a part of a naive question. We're working with Darwin Tree of Life for another project right now. So after this building all these 3D models, what do you think what is your next step as a lab or in your lab about taking this collaboration to maybe another level or
Well, the thing is, we did not collaborate it together. I used the public data
Just
already
the public
available.
database. Yeah, they also have it. They
Yes, and also like so I presented my work with Esther Gaia at the Kew Botanical Garden
oh
because I knew that she was some like her lab was making some of the data. So I was like, can I share at least what I am doing with the amazing data you produced, right?
yeah.
Um, but
Uh,
also like, sorry.
yeah, yeah, go, go, go.
But also like what I began already is to look at how much who's interacting with you. Do you have like very specific gene interactions or T gene
Mhm.
interactions? So like this is like something that would be interesting, and to see if like young transposable elements are going to be at very specific places, for instance.
That's
I
true.
think. Mhm.
So Cecile has a question, and I asked her, so I allow her to unmute her, and so she
Yeah.
can. And then Sungso has also a question. So Cecile, do you want to ask a question?
Yes, please. Alice, so you know I'm very interested into your study and very impressive work. Actually, you started with the most difficult part, which is trying to make sense of many data.
Yeah.
My question is, did you actually try? I think that's kind of following up what you just said. Did you see specific location of TEs slash heterochromatin in your 3D modeling?
I began quite some time ago, and we did not dig into it too much. But I know that some species have it more external than internal for now. But I also don't have chipseed data to cross-link with the heterochromatin, right?
Mm-hmm.
So I also like I would like to cross it with RNA6 that has been also like put available now. I think that will be like very like there is a lot to do in it, honestly.
Mm.
Yep.
So as the next step, do you guys planning to do some chipsec maybe for some of the species with?
The problem is they work with species, so some of them they still have like some material of it, but some of them not.
Yeah, I
And
would
since
guess.
I won't be in the lab anymore, I'm not gonna produce data
Oh,
personally.
okay.
I don't know if someone else is gonna do it.
Oh, okay.
Yeah.
Sungsu, do you want to ask your question?
Sure. Yeah, that was a great talk, and actually, it's a good pairing because I actually worked on fungal 3D genome organization in my PhD, incidentally. I was curious about how you think the bean shape organization is coming about, like mechanistically, and are there different sequences that are driving sort of localization in different parts of the bean? Yeah.
So this is so cool because I wanted to do a review about okay, about like fungal nuclei, because I think it's a matter of physics more than about like genome content, basically. So like there are like very cool papers that shows that the nuclei of fungi are much more flexible than the one in animals and plants. And I think that, for instance, there is a super cool video from Paris, IDBC, I think, where they show how much like the nucleus is squishing between the septa to pass by between cells, right? And I think this is very much more like how the nuclei is formed in fungi compared to animals and plants. And I think this is one of the reasons it has this very weird shape, I would say. That's my guess.
So thank you, Sungsu. I have two questions in Q&A box, and the first one is from anonymous attendee asking, are there any dedicated softwares for producing the 3D models?
Yes, and I think it's super cool. It's thanks to an amazing work from Thibaut Poinsignon. So he had a paper actually that is running a pipeline, and at the end you can have the models. So yes, and then you can view it with Molstar basically. The ref is, I can show you the ref if you need to find it. It's super nice. Honestly, I'm so grateful of all the people that worked on it before. Here you go. No. No. Here on the down left, it's 3D GB pipeline, and it's from Poinsignon 2022. Because if you
Okay.
want to test.
Thank you for that. Everett Glover asks, some pathogenic species of fungi have large phenotypic changes across their life cycles. Is it known if there are any changes in chromosomal organization across these life cycles?
I don't know. I know that it has been done in yeasts, but in fungi, I don't know. I know that there was a pathogen. It was published in 2022 where they checked different. They checked in different stages, but there were no so much configuration at the global scale, but much more like at the local scale. What is going to interact with what? But like you have to dig much more into the details at the moment. Yeah.
And then we have Serini Vasulu asking, is it possible to derive chromosome evolution based on your comparative high C data?
Can you repeat again, please?
Is
Can
it
you?
possible to derive chromosome evolution? I think the trajectory based on your comparative high C data.
Okay, so I tried so badly to have a sentinel plot. It did not work out. And we have like in this data set, I don't think so. Or like we would have to go only with the Rissolasi, which was kind of my plan at some point as well, because they are way too far away from each other. So I think if we have this kind of data in some very specific clades, I think we would have it. Especially like, for instance, in Zymoseptoria, you can see already, you can kind of track, you have the centenary and things. But with this data set, I don't think so.
Okay, as of last question before moving to our next speaker, so another anonymous attendee asked, what are the configuration characteristics of this bean shape, especially in comparison to RAB-L chromosome territories?
Well, I don't have any, like, I have some correlations. I think I have it actually, if I'm not mistaking it. So I do have differences between the phylogenetics, but not necessarily. Yes, so the bean shape, I don't know exactly how it's going to be so different. Yeah, I have to look into it, even like we have so much to dig into it, but I don't think like bean is so much different from a rubble, except that it seems to be more constrained, let's say. And also like in the rubble, we have much more like small chromosomes, like genomes, than in the bean shape in general. I think, yeah, I had it somewhere.
Oh, thank you, Alice. That was a great talk and a great discussion. And now it's our time to move to our next speaker, but which my friend Ben will introduce them. Thank you all.
Thank you so much.